morning to all of you assembled for the first session of the mid-year meeting of the Academy. I'll take only very few, not even a minute, to introduce my good friend Roop Mallik, today's morning special lecture, first lecture of the mid-year meeting he is delivering. Roop has done very fascinating work. He started off I won't go into his past history, he was in TAFR, but last 3 years ago he has moved to IIT Bombay. But he has continued to work what he has been doing and chartered into new area about 10 years ago. But what he has done is something very fascinating. Inside the cell, things are not very boring or proteins are diffusing or something like that. They have a directionality, they move around. Some of them are motor proteins, so-called nanomachines. And he has spent his, compared to me, a shorter lifetime, he has done amazing work understanding how these motor proteins function, how they go from one direction to another direction, pull each other or collaborate or fight. And you are going to hear more about the whole thing or part of it in the next 35 minutes from Roop. Floor is yours.
Good morning, everybody. Many thanks for inviting me here. I'm coming to this meeting after about maybe 10 years or so. So it's a really pleasant opportunity for me to meet old friends. And thank you for the kind introduction, Nagaraj. So as you were just told, I have moved to a new place about 3 years ago, and which is IIT Bombay. This is an evening view of the campus, really beautiful with a lot of wildlife and a lot of not so wildlife here. And this is the Powai Lake. And this is not a spaceship that has just landed in IIT Bombay, but it is just a cartoon of what cells in your body and practically in anybody's body looks like. Now, this I'm sure all of you are familiar with. It is a cut through the cell, and here is the nucleus. But what is immediately obvious from this kind of a cartoon is that the cell has many different compartments inside it, and all of these compartments have a specific function. Some have overlapping functions, but these compartments have a certain chemical composition which leads to biological function within the compartment. But these compartments must certainly talk to each other. Like this is, let's say, the endoplasmic reticulum, the nucleus, the Golgi, mitochondria. These compartments cannot completely function in isolation. They must exchange material and information with each other, and only when they do can this whole cell function, and in turn you can function. Okay, so it is really important to understand how this communication happens. It happens in many different ways. I don't have the time to cover all of it, and I don't know about all of it as well. But one of the very obvious ways is how you came here in the morning. You came from your home or from your office to this place, and you probably used a car or a scooter or a bus to get here. And something pretty much similar to that happens inside this cell. So this is a sort of a flat cartoon of what you saw here. This is a typical animal cell. There are differences, but this is a sort of a cartoon, so take it in that way. And so these are the boundaries of the cell. You would know about it as a plasma membrane, and many things in the cell are not shown here, but just what is of interest is that, let's say, here is the nucleus. So near the nucleus you have something called a microtubule organizing center. It is a complex of proteins and membranes from which these kinds of filaments come out. These filaments are also, these are very long, as you can see. A typical cell in your body from here to here could be 30-40 microns, okay? But proteins are of the order of 100 nanometers, 50 nanometers. So inside this filament, you have lots of proteins joined end to end as a polymer, okay? So this is a protein polymer, very long protein polymer. There can be thousands and thousands of the same protein joined end to end. And these polymers are called microtubules. These are not the only kinds of polymers inside cells. There are other kinds of polymers also, but just for the sake of brevity. Now, you will see a plus sign here and a minus sign here. This signifies that these microtubules have an asymmetry. This does not signify electrical charge. This signifies that these microtubules have an asymmetry. One end is different from the others in ways which I will not get into right now. But these microtubules constitute the skeleton of the cell, and like your skeleton helps you stand and walk around, this microtubule cytoskeleton also does the same kind of thing. It gives structure and rigidity to these cells, but that's not the only thing it does. These are also roadways. Now, our skeleton does not act as roadways. You don't think about things running up and down your bones, but here you have the skeleton which is in these microtubules, along which you can have a mitochondria or some bacteria or many other kinds of little packets of chemicals moving around from one place to the other. And perhaps one of the reasons that they are moving around because they want to get from this compartment to this compartment or this compartment to this compartment and so on. That is exchange of information and material, and we understand very little about how this is controlled, particularly in response to your metabolic state, in response to whether you had a drug or something or not. So these are very important basic questions. A much more visually apparent example of this is how the chameleon changes color. Okay, so if it sees something when it realizes it is going to get eaten up, it must change to the background color, and for that, just underneath the skin of this chameleon, there are cells in which once it sees something from which it wants to hide, the brain sends hormonal signals to these skin cells, and over a period of a few minutes of these, you can see. I will run this again. What happens is that inside an individual cell, there are granules of ink, pigment granules, which will go towards the center, and that is basically, you can think of these as pigment granules which can be moved towards the center of the cell or away from the center, depending on what kind of color you want to gain. So in other words, the brain and indeed many other organs can control what happens in individual cells and get things to move around in a predetermined manner, and this is important for the life of this chameleon, and this is important for your life also in many ways. Okay. Now, what we and others also have done is that you can pull out one of these things from the cell, and you can take this polymer, you can make it from, we make it from goat brains, but you can make this polymer and you can stick it to a surface, a cover slip, a microscope cover slip, and here is one of those polymers. You are seeing it under very high magnification in a microscope, and you see this, this thing here. This is, imagine one of those particles, little packets that has been taken out from the cell, and we have placed it on this microtubule, and if you look at it carefully, you will see two ends, and when you see this happening, the picture it gives you is that there are two things pulling this apart in opposite directions. That is indeed true. There is a physical tug of war of these kinds of machines. These green and red machines, you can see these arrows signify the direction in which they generate force. Such a tug of war is going on physically between. So you have to imagine these. You can't see the motors, but they are there, and you can see these being pulled. And sorry, I forgot to put a scale bar here, but this would be approximately 1 micron. The length of this would be about a micron to 2 microns. Now, so you can watch these. You can also, what you can also do, you can take a laser beam, and as this is moving, you can focus a laser beam on this here, like I'm doing right now. And if I put a lens here, the laser beam will focus onto that particle. And if this is the particle, imagine, and it comes into the laser beam, focused laser beam like this, I can, because the beam gets focused, this object acts as a lens, the photons change momentum as they pass through this beam, and when they change momentum, the photons exert a reactionary force on this particle, and because of which you can trap the particle in the optical trap. This is basically an optical tweezer. In this situation, the laser beam is going like this in this cartoon. And now when you do that, the interesting thing is that, so these are these little motors which are pulling this cargo, you can think of this as a cargo, right, that is being carried around. These motors are moving this cargo, but from these kind of optical tweezer studies and from many other biochemical studies, which I will skip right now, over several years, what we and many other complementary studies from other groups have realized that if I look at this object, if I draw a cartoon, perhaps it looks something like this. Here is the microtubule. I told you it was a polymer, so you can see these individual protein units joined end to end. And this cargo has different kinds of motors. All of them are of course not shown, but we realize that the picture is somewhat like this, that there are multiple 7 to 8 motors of one kind shown here in orange and a fewer number of motors of the other kind, the green shown here in green, which exert force in opposite directions. And there are many interesting questions which arise from this. If this is an arrangement that exists on these kind of cargoes, it may not be similar for all kinds of cargoes depending on where that thing is supposed to go. What are the forces that each of these motors generate? Typically of the order of piconewtons, but just measuring a piconewton force doesn't tell you much. You have to compare that force to something else, some biological process, some energy scale. So those kind of things are still, many of these things are unknown. What are the numbers of motors? What is the tenacity? What is the average duration over which these kind of motors can generate force? What is the mechanical compliance of this structure? How are they controlled by the cell? For example, you saw those pigment granules move back and forth. What is the geometrical organization of these motors on this surface? And something that we are really interested in, there is a lipid membrane. Just like there's a lipid membrane covering the entire cell, each of these cargoes here, there's a lipid membrane covering it. There's a membrane here. And how does this motor attach to this membrane? What are the rules and principles of that? And how does that matter also for your health? These are things that we have been interested in, and I will try to touch upon that a little bit. I will not be able to go into details of too many things, but just to give you a flavor. Now, here is an object which is supposed to move. You see these teams of motors, one trying to take you toward this side and one trying to take you to the other side. How is the balance of their activity control to decide where this thing will end up? Now, there have been studies over many, many years, decades actually, where people have isolated a single motor and done these kind of optical tweezer studies and other kinds of biochemical studies and understood a lot about these motors. And one of the things that, some of the things that you can have been very precisely characterized is, so you can think of these are enzymes, right? These are enzymes in which a chemical cycle is coupled to a mechanical cycle. So when they burn energy, and energy is in the form of adenosine triphosphate which comes from your food, when they burn energy in a cycle, there is a mechanical change in the protein which makes this protein walk like I am walking. Okay? You have this picture where these things are moving. Now, each of these motors, they have an on rate and an off rate, just like an enzyme. If you think of it an enzyme, it has a particular rate at it which will get onto this and start generating force, and it will generate force for a while, and then it will finish its cycles. Okay, so that's an off rate. Now, what we do know is that there is an arrangement where there are a few kinesin motors like this and significantly larger number of dynein motors, which you see in this. This is the dynein. Dyne comes from the word dynein comes from the word dyne, and kinesin from motion kind. Okay, so now what we realize that given the on rates and the off rates, if you have this kind of an arrangement, you can never get this object to move in this direction. Okay, because there are so many of these motors with a given on rate, one or the other of these will always be attached. These, these will never get a chance most of the time. Okay, but you do require this asymmetric arrangement where these motors are much weaker. The orange motors generate about 1/5 of the force that these green motors generate, and so having 1 or 2 here and 6 or 7 is required for. Otherwise, there is no competition ever. Okay, so but once you have this force balance, these will never let go because any of them can attach, and the effective on rate becomes very high. Okay, so then what we realize that, as I told you, that these motors pull against each other. So what we realize is that to now, yet it's not that these cargoes always move in this direction. They move in both directions. So how does this green motor ever get a chance is the question. So what we realize is that when this tug of war happens, I showed you this membrane getting stretched, right? So these are pulling against each other. Then this guy, whenever it is attached, it pulls strongly against these. And this dynein motor, which others have also kind of speculated, enters into something called a catch bond like state. Okay, so basically you can imagine two links which are if they are loose, then if you pull them gradually apart, then you can separate them. But if you pre-stress them like that, then they will bind and you will have a catch bond like effect here. That is known to happen right here where this dynein meets this microtubule, and there is a catch bond activated when these pull in the other direction. Well, that was known. What we proposed based on the our experimental data, where we pulled this optical tweezer with this optical tweezer, is that once the dynein recovers, once the dynein comes out of a catch bond state, it goes into some kind of an off pathway state. There is an energy minimum probably, and it can't get out of this off pathway state for a while. If it can't get out of this off pathway state, it cannot engage with the microtubule, and that is when kinesin gets a chance. So if you shine a laser beam like this and this object is moving back and forth, here you can see that this is only dynein driven motion. Imagine that this is only going this direction, and I want to activate a catch bond in these. What do I have to do? I have to pull them forcefully. What I simply do is that when dynein is walking like this, it is walking on a cover slip, I pull the cover slip towards the front, and I'm already pulling it with this optical tweezer at the back. So I pull the rug from under dynein. You can think of it like that. So this data is typical of what you get in an optical tweezer. This axis is time. Okay, so as time passes, this cargo tries to move away from the center of the optical trap, and you have to imagine the optical trap as a spring here, okay, that I've tried to show as a cartoon. When you move away from the center like this, imagine this object going this way, the spring gets pulled. Okay, as the spring gets pulled, the spring pulls back, which is the optical trap pulls back, and somewhere you reach a stall, something like a balance situation, and by measuring this distance, you can calibrate this optical trap, you can measure how much force is being generated of the order of piconewton. But what we did here is that once things kind of reached an equilibrium, we pulled the rug from under dynein like this, and then you do this, okay? This is where you induce a catch bond on the dynein, and then you watch how long does it take for the next event to happen. That is the recovery time. And when we do such experiments, what we find that when these times, the ones shown in blue where you have induced this kind of a state, forceful state, the recovery time is longer. Okay. Again, I don't want to spend too much time on this because I lost some time, but you can now do this kind of experiment where both motors are active. Okay, so here you see this is the center of the optical tweezer. Now kinesin can pull you this way or dynein can pull you this way, and these measurements are done at several kilohertz sampling frequency with few nanometers precision here, 4 or 5 nanometers, so you don't miss too many events. Now what you do here is that you induce this, you pull the rug from under dynein, let's say here, and if whatever I was saying is true, then the next event has a higher probability of being kinesin, right? Because dynein was inactivated by pulling in this manner, and then the next event, kinesin should get a chance, and that's actually what from our preliminary measurement, we are still doing these experiments, the reversal probability, if it is a stall is where you have not pulled the rug. The balance is coming out by itself, but when you exert this additional force, we call it super stall, you now see that the chance that you will have a kinesin event after dynein increases. And this kind of a situation where you have this tug of war inside cells and probably this kind of a catch bond happens in the context of many kinds of cargoes, and those are things that, just to give you a flavor of that. All right, so my title is Moving Fat on the Move. So I wanted to talk about something else also. So my lab has two kind of parts which connect and sometimes do not connect. And so where we study these kind of biophysical properties of these motor proteins and their implications to cell biology. I gave you a flavor of that just now. We have also been, so I told you just now that these cargoes that are being moved around, let's say a mitochondria or a bacteria or a virus which just got inside a cell, it has to go to a particular place, let's say the virus to multiply, okay? So we have to, and that it has to be, where it goes is decided by the relative activity of these two kinds of motors. So we really need to understand these things, how these motor proteins are, let's say, hijacked by these viruses or bacteria to replicate. But most of the cargoes which are inside cells, let's say this is the interior of the cargo where you have your proteins or whatever you have here, is covered by a lipid membrane. And you can imagine that the outside of this cargo, which is the contents, this is the cytosol, which is the stuff in the cell, is watery, right? And the inside is also watery. So you need to separate this watery compartment from the outside compartment, and that you do by making a membrane around it. And because both sides is water, this membrane must be a bilayer membrane with the hydrophobic parts, fatty acid chains inside, and the polar head groups on the outside on both sides. Otherwise, it will not be stable, okay? Now, a lot of people, actually this is a very less studied problem. How do these motors bind to these membranes? And when they bind, how are they organized on this membrane? And how do metabolic signals, how do many other cellular signals control the rules by which these two bind? And that is absolutely essential to understand if we are to understand which motor will bind and be active at what time, okay? So this is some motors shown bound to this membrane. There are, of course, many other proteins which we are not talking about. But what we have been interested in is another kind of cargo. So I just talked to you about mitochondria and virus and things, but do you have fat deposits in your body? Okay, on practically every organ in your body you have fat deposits, and that fat gives you the membrane to make these kinds of lipids, to make new cells, to make new things. It is also the most concentrated energy source in your body. Okay, these hydrocarbon chains have a huge amount of energy, much more than proteins actually, or carbohydrates, given the mass. That energy is stored here. Now, if this fat deposits in your body are called lipid droplets. Okay, so they have lipid inside, and they are again going to be covered by a membrane. And yeah, so this stuff that you eat goes into these lipid droplets. And now if I take a lipid droplet here, it will be fundamentally different because the membrane covering it cannot be a bilayer. Okay, the membrane covering it must be a monolayer because the inside is fatty. Okay, so these hydrocarbon chains must be exposed to the fatty part, and the outside has to remain polar because there is water on the outside. Okay, now this immediately means that the rules by which proteins attach to this membrane are going to be completely different. Okay, because what attaches to a bilayer membrane may or may not attach to a monolayer. This is a monolayer phospholipid membrane, and this is something which we have been really interested in. What are the rules by which proteins or other factors differentiate between a bilayer membrane and a monolayer membrane? Now, why is this important to understand? Why should you care about lipid droplets? So, I believe that this membrane on the lipid droplet is both the lock and the key to your metabolism, okay? Because this is where you store energy, and energy is a fundamentally important part of your metabolism. So what comes and attaches to these lipid droplets are not just sitting around in your body. They are extremely dynamic. They are going to different places. They are giving off fatty acids. They are taking in other things. So they are supplying energy and material depending on the demands of your body. Let's say you don't eat today, okay? How are you going to, if you don't eat for the next 6 hours, these lipid droplets in all the kinds of cells in your body, particularly your adipose tissue, are going to get eaten up, and that energy you need. Okay, that's why you get thin when you don't eat. All right. So the proteins which attach to this membrane can be of different types. These lipid droplets, fat deposits in any typical cell type in your body, they actually emerge from the endoplasmic reticulum. So what happens is that the endoplasmic reticulum of many different types of cells is long, extensive membrane, and there are enzymes on this endoplasmic reticulum which synthesize molecules of triacylglycerol. Triacylglycerol is 3 fatty acid chains bound to a glycerol molecule. That's a neutral, effectively it's neutral, electrically neutral. Because it is electrically neutral, you can store large amounts of this material in a small volume. That is the fundamental basis of storage of energy in your body. You cannot store charged particles in a compact manner because they will simply repel each other, okay? So you basically esterify these fatty acid chains to glycerol to make it a electrically neutral molecule, and you can store lots of it here. But once you have stored it, you don't want to just let it be there. You want to access it when you need. So there are these enzymes which must come and bind to this, lipases which chew up this fat and release this fat and use it in different ways. So, so therefore, it is important to understand the biophysical properties and the biochemical properties of this lipid membrane, which allow lipid to be stored or used depending on your metabolic requirements. And as you know, some of the biggest problems in health are because of metabolic disorders leading to different kinds of conditions. And so these are really important questions to understand. What is the packing of these phospholipids is very important to understand how these proteins will embed on this. What is the surface tension here? What is the curvature here? These are things which one needs to understand and connect it to physiology, connect it to what happens to the animal. These are really important questions. So in this context, we are doing some other things, but we have been very interested in what happens in the liver. Okay. Now your liver, it does many things, but one of the major functions of the liver is to balance lipid overall in your body. Okay. You have a certain content of fat in your blood. That fat, cholesterol, all of this is controlled by your liver, because the liver also makes cholesterol and glucose, as you know. So coming to fat, the fat that you eat in your burgers, okay, that comes from your diet, and that fat is secreted out from your intestine and eventually goes to your liver. Okay, the fat in your adipose tissue, that also, depending on whether you have eaten or not eaten, will ultimately go to your liver. And the liver takes this fat input from all places in your body, integrates it, stores it into these lipid droplets inside the cells of the liver, and releases this fat out into your bloodstream in a very controlled manner. You have to thank your liver for your health. Okay, and when this fat is secreted out from your liver, it makes up the serum triglycerides that you see in your blood report. Okay, high serum triglycerides, you can understand, is a cause for serious worry if that happens to you. Now, so let us focus now on these lipid droplets inside the cells of the liver, okay, and see what it means. What is the connection to what I told you a little earlier. Now, this is the lipid droplet which we isolated from the liver of a rat, okay? And when we take this lipid droplet, you remember that earlier movie I showed you where I had a microtubule stuck along this on a cover slip and something was moving. Here is a lipid droplet which I have pulled out from the liver. And if I put it on the microtubule, you can see it moves so beautifully. This is in real time. This is not sped up, okay? So this velocity is a few microns per second. And it moves beautifully from one side of the microtubule. You can't see the microtubule, it is in low contrast. And so now you have to imagine these lipid droplets taken out from the liver of a rat, and put them on microtubules, they move in this manner. And what we realized is that when we do this experiment with rats which have been eating happily, all of these lipid droplets move in a particular direction. Okay, they're all driven by the kinesin motor, as I've shown in this in this cartoon. Then the obvious question is, why are they moving? Are they moving like this inside the liver as well? Or is this something that happens, just pulled it out, so it's all wrong, okay? by many experiments, we actually verified that this happens inside the liver also. And to cut a very long story short, I will show you, try to explain to you what happened. This is a little complicated, but imagine that this is a cell in your liver. Okay, this is called a hepatocyte. Okay, your liver is like a giant sieve. It takes in, drugs or fat, all kinds of things from one side and filters it through these hepatocytes and send it out the other side to circulate in your body. Okay, crudely you can think of the liver like that. And inside the liver, what is happening is that when your insulin levels are high in your body, okay, that is mean when you are nicely eaten and your insulin levels are high, then this insulin activates a cascade of proteins, which I won't go into right now. And one of those proteins takes the phospholipid on the surface of this lipid droplets and converts it into a different phospholipid. This is phosphatidic acid now. Now, phosphatidic acid, when it is present on this membrane, it has a slightly negative curvature. Means that the head is small, the legs are splayed like this. Okay, so once you generate phosphatidic acid in the membrane, it generates a local curvature in that area. And now, because of that local curvature, two things happen. One is that the kinesin motor comes and directly binds to this phosphatidic acid and carries it along this microtubule towards the periphery of the hepatocytes. What is the point in going to the periphery of the hepatocyte is that the enzymes which will chop up the fat inside, which you want to send out into blood, okay, remember, to make out your serum triglycerides, to make your body running actually, that is present here. So you use the lipases present in this smooth endoplasmic reticulum, which is present at the periphery of the cell, to chew up these lipids and repackage them into something called very low density lipoproteins. These very low density lipoproteins are going to be secreted out from the liver. Now, it is also interesting to note that when this lipid droplet reaches this smooth endoplasmic reticulum, if you come here, imagine this lipid droplet, this one coming, and it has to fuse somehow or interact with this membrane. That is not going to be easy. There's an energetic barrier to that. Okay. How does that happen? We believe it happens because the phosphatidic acid, which was generated on this, induces a negative curvature here. Okay, so you can form these neck-like regions so that the lipases can go from here to here. And if you block this process, you may have very little, serum triglycerides in your blood, and your energy levels will go down. So these are really interesting questions. Now, once we figured all this out, as you know, there are people with high serum triglycerides. This keeps coming up, which leads to high cholesterol and these kind of ailments, dyslipidemia in general. So can we reduce the amount of triglycerides secreted out from the liver in some way? There are other ways of doing it. We are also trying to find a different way of doing it, where if you can probably control the transport of these lipid droplets to this location, then can you reduce the amount of VLDL, serum triglycerides in your blood, and will that help, maybe that can help patients. So so how do you do it, okay? Now remember that this kinesin motor, it carries lipid droplets from here to here, but it does 100 other things inside the cell. So if I go and block this kinesin motor, that is not the way to do it, because that would be disaster, okay? You have to find some way to block the activity of kinesin specifically on lipid droplets and not on anything else. And that is where we figured out that the fundamental manner in which any protein binds to a monolayer membrane must be different from a bilayer membrane. So it may be possible to block the binding of kinesin to this lipid droplet and not block the binding of kinesin, let's say, to the other things inside the cell. And if we can do that, maybe that would help. So we are testing this hypothesis, and we are testing it in cell culture, and we are testing it in an animal model. Both experiments are in progress. Shubham and Archishman are doing this. So what we can do is that there are other things. I'm just giving a very small glimpse of this. So 5 more minutes now. Yeah. Okay. So you can take a bead essentially. It's a plastic bead which has polar groups on the surface. And if you add phospholipids to it, because this is polar, you will make a bilayer membranoid. But if I take a bead which is not polar, which is hydrophobic, and I add phospholipids, I will make a monolayer membranoid. So this represents a lipid droplet, and this represents other kinds of organelles. And now, when I add so here is the kinesin motor. You know, you can compare it to a human being walking like this. Here is the kinesin motor. These are the places where you hydrolyze ATP. And this part, just like it is to carry a cargo, must bind to a lipid droplet or to whatever other cargo. We and others figured out that the mechanism by which the kinesin motor binds to bilayer membranes like this uses different parts of the kinesin. To bind to a monolayer membrane like this one or a lipid droplet, it binds to this region called the kinesin tail domain. This you have to remember. So if I now, so let's say I take this kinesin motor and I make it green. It gives green fluorescence, and I throw equal amount of motor on this kind of things or this kind of things. What you see is that so I make this. Sorry, I said wrongly. This part I make and I add a fluorescent tag to it. This is the kinesin tail domain. You can see that hardly anything gets onto the bilayer, but the monolayer has so much of the kinesin tail domain. This suggests that this part preferentially binds to a monolayer. So if I now have a proper kinesin motor bound to a cargo, and I make lots of these small peptides, okay, and I throw it on this, just by simple mass action, these small in this region will come and bind here, and it should displace the entire motor. And this is one crude way of doing it. We are thinking of other ways also, and it actually works. That if you have the membrane, this is the amount of kinesin, and when I add the kinesin tail domain, there is hardly any kinesin left on the motor, and we have checked this. I will not go too much into this. The most important thing is that would this actually work in an animal? Okay, if you ask me this, we don't know the answer, but we'll tell you what we have done. We're collaborating with Dr. Shailaja Nair to use zebrafish. Zebrafish are small fish. They are a fantastic model system for microscopy. They are vertebrates. Okay, so many of the things that happen to you happen to zebrafish also, including lipid transport, lipid balance, lipid homeostasis. So what we have done here is that we have taken a red lipid. Okay, it's a fatty acid chain, red lipid, think of it, and you simply, this is very interesting, some of you may want to use this, you can buy eggs from the market, and you mash up the eggs, and you do a centrifugation, you can make liposomes from eggs. Okay, so which has all these phospholipids, everything you have, and now when you make these liposomes, we simply add some of this kinesin tail domain which I told you, it gets incorporated into the liposomes. Okay, now when these larvae are swimming around in the water, we feed them the liposomes. The larvae will happily eat the liposomes, okay, because it's energy for them and phospholipids, and in that process, the fatty acid, the fat that you have delivered to these larvae also enters the body. This is a way of delivering this kinesin tail domain peptide. So what we find is that in a normal fish, okay, when you deliver this fatty acid, the red fatty acid, it goes into these parts of the body. What is happening is that the fish is eating it, the red fat is going to the intestine and to the liver. From the liver and the intestine, it is getting secreted out all over the body, and you see that happening. But when I add this kinesin tail domain, you don't see that happening. Okay? When I add a mutant version which is not supposed to work, again you see recovery. So this is suggesting that the kinesin tail domain is able to block, at least to some extent, fat secretion from the liver into blood, and we have to take this forward. We will test this. We can test this biochemically also on large samples. I will not get into this. I will take 2 minutes just to tell you about something else, 2 different directions. Maybe I'll not use the slides. We are also very interested in what anti-cancer drugs and antidepressants mean for lipid droplet biology. And why these? Because many of the anti-cancer drugs that people have to take, unfortunately, are so-called tyrosine kinase inhibitors. Okay, they inhibit kinases, and these drugs are extremely hydrophobic. Most of these drugs are taken orally, and they have to go through your liver to get anywhere else. Okay. Now, if your liver has lipid droplets, the conjecture is that most of the drug just simply gets trapped in the liver. Okay, because they are hydrophobic and they get trapped in the hepatocytes. And as you know, about 95% of the drugs fail because they cause liver injury. Okay, so that might be a reason for why this happens. We are investigating this by other means and trying to find a molecular mechanism or a physical mechanism by which this happens. And we are also looking at some antidepressant drugs, why they cause obesity, and we think they do so by activating a particular way in which mitochondria and lipid droplets interact in cells, in hepatocytes. I will skip all of this and I will go to my last slide, where I would like to, before I end, tell you that a lot of new things are happening at IIT Bombay. Some of the things are listed here, which very high-end microscopy facilities, next generation sequencing, a digital center, Koita Center for Digital Health. I would encourage you to come by sometime if you are around in Bombay. I would like to acknowledge people who fund us and collaborators with who make all this worthwhile. And of course, the people who do the work, and this is last year. And a lot of what I told you was done by Dvitiya. I could not talk about what Shayantan and Apurva are doing. And that's Pauline who has left the lab, and Archishman. Thank you very much.
I wanted to know how the rate of movement of the cargo is controlled by dual action of dynein and kinesin. What are they doing to control the rate of the movement?
There can be many answers to this, and some of them we know, some of them we don't know. One is, let's say a certain other protein comes and attaches to a kinesin motor and increases its on rate or reduces its off rate. Okay, such proteins are known. If they do so, then you would increase kinesin dependent motion compared to dynein, and it could happen the other way also. That is one way. Another way could be the lipids on the membrane, depending on your metabolic processes, could control the geometrical organization of the kinesins and the dyneins on the surface, because you can imagine that it's a curved surface and there is a straight line underneath it, so only a limited part can access this. So if you change the membrane organization, the density, then you can profoundly affect things. So these are two ways. There are possibly many other ways which I don't know of.
Thank you for a very nice talk. So regarding the first part of your talk, where you had this lot more dyneins. Yes, yeah. I'm mixing the kinesin.
Yeah.
Is there any evidence from your experiments that these motors are in lockstep? The on-off rates are in sync?
It's very hard to answer that question. To some extent, yes. So I think what you're trying to say is that do they share load equally? Yeah. So there is evidence that they don't. But in our particular condition, I couldn't go into those details. We believe that the dynein motors are arranged into very tight, small clusters because they assemble on lipid rafts on that membrane. And if they are in a tight cluster, basically their attachment points are very close to each other, then they should be in lockstep. Okay. Now, we have some evidence for this, but I won't put my neck on the line for this. Yeah.
I just wanted to check whether you have that dynein-dynesin thing. One is more in number, smaller the force, larger the recovery time. Possibly Poisson distributed each one of them.
Yes.
On the other side also Poisson distributed large force.
Yes.
Would there be a statistical answer to the question?
There is a statistical answer. We have those. We are working with people. We also do these kinds of simulations. If you're interested, we don't know the entire answer, but I can discuss this with you.
Are the ring roads?
I can discuss this with you.
Wonderful talk. I have a question regarding the optical tweezer.
Yeah.
So there's a certain amount of heat that usually gets generated from the tweezer.
Right.
Does that affect the mechanism?
Yeah. So typically the temperature change because of that. We and others have measured it. It's a few degrees centigrade. Okay. Given the laser powers that we use, it's a small perturbation, but it's not the end of the world. Yeah.
So if there are no other questions there, I have one quick one. Determines this asymmetry that dyneins are on one side and kinesis on the other side?
We don't know that. That was just a cartoon. We don't have the resolution to say that really. But given the way in which the force is generated, you presume that. Yeah, I should have mentioned that clearly. Yeah.
So thanks very much for very, very nice talk.